director, one of 2 staff people we now have, which is a big deal for Monterey Audubon in the last year or 2. Anyway, so thank you all so much for your patience. I'm really excited to introduce Dr. Kelly Barr presenting tonight about burrowing owls, which is one of those charismatic, amazing little critters that we all really want to see, kind of obsess about, and they're kind of in decline. Get to see them sometimes in the winter, like at Paula Corona Regional Park, or I saw one once on the rec trail behind Costco in the ice plant. But kind of few and far between, but really cool little guy. And we're excited to hear more about it. Dr. Kelly Barr is a professor over at CSUMB, Cal State Monterey Bay, just up in Marina, or Seaside, I guess officially is the address. And it's just really cool because I went to school there and there weren't a lot of wildlife classes. It's kind of just been in the last 10 years, they've got a wildlife ecology professor, Dr. Jenny Duggan, and then Dr. Derek Bergsma. He was doing like a summer month-long field ornithology class, but now we have a bona fide, like semester-long ornithology class at CSUMB, taught by Dr. Kelly Barr, and it's just the coolest. I wish I could go back and do it again at CSUMB. Anyway, so I just wanted to thank you so much for coming. Thank you all for your patience, and yeah, take it away.
Really never talked on the microphone before. Can you guys hear me? Is this good for everybody? Okay. A little farther from my mouth? Okay. Good evening. Thanks for having me. My name is Kelly Barr. Thank you for the introduction, Amanda. I am going to be talking to you guys about burrowing owls, but I'm also going to tell you a few other things. This burrowing owl research part of the talk is bracketed by a little bit of my bio, a little bit of my background, and I'm also going to talk about a little bit about the research that my students, many of whom are here today, did in the first iteration of, I'm calling it bird biology, it's just to differentiate it from Jarek's field ornithology class. So there was supposed to be a audio file here to get started, but we're just going to wait to that. And first thing we're doing is getting stuck. Did Chris take off? Oh no, there we go. Okay. All right. So, gave it away. So I always start off my talks by pointing out how unlikely it is that I'm the person talking up here today. And for the Audubon Society, that is doubly so. Number one, I am from a tiny little town, one stoplight town in eastern North Carolina called Chocowinity, North Carolina, population 600, or as I joke every time, 599 because I am here, and it's one of those kinds of places that nobody ever leaves. Number two, I did not want to be a bird researcher when I was a young person, thinking about college, going to college. I wanted to be a marine scientist. So with that in mind, I ended up going to the University of North Carolina, out of Chocowinity, and getting a degree in environmental sciences and engineering with a minor in marine sciences. And I did that only because marine sciences was not an option as a major as an undergrad. So that was the way that I got the closest possible route towards marine sciences, the closest matching degree. Now, out of undergrad, I acquired some genetics lab technique that I learned in a job that I had during undergrad and right after undergrad. And based on those skills, I was able to get a job down at the Smithsonian Tropical Research Institute in Panama. And at 23 years old, I got onto a plane for the first time. Again, one stoplight town, 600 people, parents didn't graduate high school. I was not a world traveler when I was growing up. Went to Panama, and I was in Panama for a year doing marine sciences research. I was working in a lab, spending a lot of time going into the Rocky Intertidal, to the coral reefs, and all the wonderful marine sciences things that marine biology things that Panama has to offer. Never once did I stop and look through a pair of binoculars in Panama, a place that people go from all over the world to see the birds. That is a crime I need to correct at some point in my life. I have to go back at some point in the future. Now, when I was an undergrad, when I was working at Smithsonian Tropical Research Institute, I had the good fortune of going to talks like this, sitting in the audience like you guys, and hearing about all this amazing research around the world. And I got inspired to become a researcher, but also knew that I needed to learn a lot more. So I decided to come back to the United States, and I found a amazing project at the University of Louisiana Lafayette. If you guys don't know, that's referred to as ULala, UL Lafayette. I came back to work at Ulala to work on a project studying the effects of sea level rise on coastal fishes and using genetic techniques to understand how selection is changing for their populations. And I was very excited about that project. It was marine biology, you know, that's what I wanted to do. So how this happened, okay, this is getting caught up again. Okay, we'll wait for that to switch over, and I'm not going to hit the button 3 times. So when I got to Louisiana Lafayette, the start of my master's degree was delayed a little bit for personal reasons, and some other students had already been picked up to take that coastal fishes project. So instead, my advisor came to me and said, "Hey, we could probably squeeze you in on that project, but we have this other project, this fully funded project to work on endangered species in Texas, endangered birds in Texas." And is the thing going to switch over? And in the back of my mind, I thought, well, it's Texas. It's birds. I just spent a year in Panama, and I didn't seem to pick up the love of birds then. Yeah, this is stuck again. I'm hitting that, yeah. That too. Let's try this. Chris. Let's see. What if I hit that slide? Oh, I figured out a hack. Okay. There's another button I could hit. So I said yes. I said yes because it was a fully funded project. I said yes because it was endangered species. I said yes because I knew that in my life I wanted to work in species conservation. So I knew that this would be a good route to get there. I was really unsure about the birds. Within weeks, I fell in love with birds. If you guys haven't been, Texas is an amazing place to visit as somebody who's interested in birds. It's a major flyway for migrating birds. The bird diversity there is incredible. It is another place that people come to from all over the world to see birds. So I worked on these 2 species, the black-capped vireo and the golden-cheeked warbler, and I got to see a whole lot of the Texas hill country while I was working on those birds. Now, another thing that I learned when I was working on that master's degree is that if you have birding skills, if you're able to identify birds by sight and sound, if you're able to catch birds, in my case, put bands on them and take a little bit of blood from them, then there are jobs for you all over the world. Or as I tell my students, there are jobs for you if you're willing to not get paid anything and sleep on the floor. When you're 26 years old, I think I was at the time when I graduated, I was willing to go sleep on the floor. I wanted to travel. I wanted to get out of Louisiana. I wanted to get out of Texas and get out to use those skills. So I got a job down at CSIRO. CSIRO is sort of like the catch-all government agency that does everything that the U.S. Geological Survey, Fish and Wildlife does here in the United States. And I was doing a project there, doing radio telemetry. Let's see. Is that picture going to come up? So that's what that looked like, walking around the outback from sunup to sundown for 6 days a week for 4 months, seeing amazing bird life. Not just seeing amazing bird life, handling amazing bird life. We had to catch and ban these birds. That's 27-year-old me, students who are here. Back before the hair went gray, of course. That is a laughing kookaburra. We caught several of those. I look terrified because that is a big bird, the big beak. And mostly I just didn't want it to get out of my hand. This is my favorite bird in the world. It's either Australia has really bad bird names. Superb or splendid or something like that, fairy wren. Instead of giving them like sensible names, they just have these descriptive names. That's a turquoise parakeet. All amazing birds that I got to handle. Got to see amazing birds like the rainbow bee eater, the rainbow lorikeet. I can't remember students that are here in bird biology. When you guys asked me what my favorite bird is, I think I told you guys that the willy wagtail is my favorite bird. Look at that eyebrow. They lay their eggs essentially out on the ends of these tree branches, just out in the open, and then they just mean mug you if you try to approach them. So it's such a really cool bird. So I did this job in Australia for 4 months, And with some friends that I made doing that job, and I purchased a small SUV and spent another 8 months traveling completely all around Australia. And Australia is expensive, at least back then. Gas is even more expensive than even California. So I was broke and I needed a job. So I came back to the United States, and initially I was sleeping on my brother's couch in Atlanta, Georgia, and I saw this job come up in California at Hopkins Marine Station. Hey, isn't that kismet? So I applied for this job at Hopkins Marine Station, getting back to the marine world. Back in 2008, I came out and interviewed and scored the job with Steve Palumbi, whose lab's just right over there. I really feel like his life's come full circle today. And it was really fun work. It was really fulfilling because I was getting to do a lot of travel, getting to see a lot more of the world, the entire Oregon coastline, a lot of California. And I thought it was going to be like Australia. I thought I was going to come to California and only be here for a year and then take off and go to somewhere else. A year after being here, I met my wife on Alvarado Street. And she's from Marina, so we live here. Also, kind of love being here. But that's not the full story. The full story is I was working at Hopkins, and I was collecting data, handing it off to Steve so he could write papers, and it was fulfilling. I liked the work, but what I really wanted to do was my own projects. I wanted to do the full scientific work, from applying for grants to collecting samples again to writing papers. And I saw a job advertised down in San Diego at the U.S. Geological Survey that just happened to be seeking somebody with genetic skills and skills to handle birds. So, hey, that was me. So I applied for and got that job and dragged my wife away from her family for 5 years to go live down in San Diego, working on more birds. So I worked on the cactus wren there, the California gnatcatcher there. I had a short foray outer birds, worked on California red-legged frogs as well. Really fun field work, going out at night into waterways and ponds to catch those guys. But when I was with the U.S. Geological Survey, I had two problems. Number one, the world was changing really fast. There were these advances that were happening in genetics that meant that the skills that I had were becoming a thing of the past really fast. So I needed to learn new skills. We were entering the world of genomics. You guys can send your DNA off to get your genotypes done for $100 these days. That was not the case when I first started doing work in genetics. So I wanted to learn those skills. But I also had a desire to go get a PhD. I kind of mentioned it offhand earlier. My parents didn't graduate high school, much less go to college. So I really wanted to have this advancement for my family in the future. So I found a home for myself at UCLA. Let's see. Oh, this button's not working now. It's like different buttons work at different times. So I found a home for myself at UCLA and developed a couple projects working on birds. The tricolor blackbird, one of my focuses of study. One of my field sites, well, two of my field sites was up here, Laguna Seca, and over in Carmel Valley, there's a really small pond on private property that I got to catch some tricolor blackbirds at. But also burrowing owls. So let's talk about burrowing owls. Oh, hey, I meant to say this at the beginning. Typically when we do these talks, I talk a whole bunch, and then at the end, you guys ask questions. I want to encourage you, if you have any questions as I'm talking, just raise your hand. My students will report that when I teach class, I try to stop talking every 20 minutes or so, at least. At least. So let's talk about burrowing owls. So burrowing owls are spread throughout the Americas. If you throw a dart at that map, you'll probably hit a new subspecies. There's about 20, they're split up into about 24 subspecies. And the ones that I worked on are the only subspecies that has migratory populations. That's here in Western North America. Hypugaea is the name of the subspecies. Burrowing owls are a little complicated because throughout the rest of that range, they're resident, they breed and winter in the same place through the annual cycle. Western burrowing owls are more complicated. they have both migratory populations, whatever that color is at the top. I'm colorblind, and I showed Lunar what these colors are. Whatever that migratory, that's why I got an arrow pointing right there. So that part of the range, they're only there during the summer when they migrate there to breed, but then they migrate back south during the winter. But in that intermediate range, there's a whole bunch of resident owl populations. And previous research, kind of with older techniques, less data-rich techniques, suggested that there were no differences between any of these populations, that the burrowing owls were all sharing genes, alleles, I should say, across their range. So it was an interesting system to get involved with because of these multitude of complications.
What was the time frame of their migration?
Well, it's a typical migratory breeding cycle. So in the spring they migrate north, and in the fall they migrate back south. So it just takes a couple weeks.
Oh, okay.
Certainly not an extended period of time. Yeah, I'll mention that in a second. Now, throughout this range, they have, as Amanda mentioned, there are varying levels of protections for the owls because many of their populations are in trouble. That really complicated map on the left there portrays the fossorial animals that they rely on to make their burrows. This is an owl a bird that is poorly named. It's called the burrowing owl, implying they can burrow, they cannot. They should be called a burrow owl. They have to have other reptiles or mammals to make their burrows for them. So one of the problems that the burrowing owls have is many of those fossorial animals themselves have been in decline. So they've lost habitat. On this map, this hashed out region are places where burrowing owls used to be, and they are no longer. And this is a study from 2018, so that probably has expanded a little bit, that area where they've been extirpated. The other problem, there are many problems that burrowing owls have. Outdoor cats is a problem for burrowing owls in urban areas. This is an image of the turbines up in Livermore and the Altamont Pass. In the Altamont Pass alone, I don't know if it's still the case, but when I started this project 10 years ago, 500 owls a year were killed by those turbines. So it was those turbines were really nasty for them. There are species of concern in a whole bunch of states, including California, and they are currently up for potential listing under the California Endangered Species Act. And I support that listing. Just to portray that a little more, this is from the petition to list the owl that's on the California Fish and Wildlife website. So this portrays where the owls have, in better detail than the other map, in hashes where the owls have been extirpated in the state of California, where their populations have completely gone away, and places where their populations are really, really low, near extirpation. So they're suffering in a lot of places. In 2003, it was estimated to be around 8,000 to 10,000 owls. 2023, when this petition was first submitted, there were about 6,500 breeding pairs of resident breeding owls in the state of California. Now, this doesn't tell the full story, because 4,000 of those breeding pairs are in one place, down here in the Imperial Valley. And put a pin in that. We'll talk more about that in a minute. So to do research on the owls, they're spread out throughout all of Western North America. Catching them is difficult. It's arduous field work, really fun field work, but arduous in terms of time. So I had to work with a lot of partners to get samples, and I'll just put up a whole bunch of images now to represent a bunch of partners that helped with getting samples for this project. But with those samples, I wanted to answer a couple of questions. First of all, we talked about how they have this complicated migratory breeding behavior, this migratory populations versus resident populations. So I wanted to try to understand how that might affect genetic structure, which means how much movement essentially there is across their populations, between their populations, how it affects genetic diversity, which is something that I care a lot about because that says something about how healthy their populations are. And then is there any sort of evidence that we could pick up using this data that suggests there's something going on in terms of natural selection that differentiates those resident breeding owls versus the migratory breeding owls? Any questions? Mostly I just want to get a drink. Yeah.
I always kind of thought that if something's migratory, it's inherently going to be more at risk of decline. Is that, did you go in with that assumption as well, or is that a crazy idea I have?
Not a crazy idea. Certainly with the burrowing owls. So what's happened is their range is receding from the north, from the east, from the west. And if they're resident or they're migratory, then their populations are in decline on those fringes.
Thank you.
So what I did in collaboration with all these partners was to collect samples from throughout the range. Everywhere on the map, there's a point. That's where I got some blood samples of owls, mostly blood samples of owls. There's a couple other things to point out here. Anywhere that there's a triangle on the map, that is a population that we know to be migratory. So of course, we sampled these owls during the breeding season, so we would be able to differentiate whether they're migratory or resident. Anywhere there's a circle on the map, those are resident populations of owls. So note, it's mostly California. The resident owls are largely in this state where, as I pointed out before, they're in great decline. So keep those triangles and circles in your minds and what they mean. Yes?
The Channel Islands, and I remember seeing them on the Channel Islands, but it was 50 years ago.
So my understanding is with the Channel Islands, they do get wintering migratory owls. They get them out on the Farallones. I'm trying to turn that into some sort of boondoggle to get myself to the Farallones at some point. My understanding is that all those owls that were resident breeders out on the islands are extirpated.
Gone?
Gone. Yeah, those populations have gone extinct. So the triangles, migratory breeding owls, circles, resident breeding owls. I'm also going to put some squares up there. So when burrowing owls migrate back south, some of them overwinter alongside of resident breeding owls. So in many places in California, if you go out in December, you might see resident breeding owls, but you also might see some migratory breeding owls, migratory owls that are overwintering. So they will overwinter right next to the resident owls. Yeah.
Climate change?
Put a pin in that. Possibly. There's something to talk about with that. No, they overwinter because it's good habitat. Because there's conspecific attraction. If you're migrating and there are other species that are the same species as you know that's good habitat that you can survive in. So that is the case in many of the populations, resident breeding populations. So I put squares up there because these are two populations that both the data that I collected and anecdotally other researchers suggested that something's happening with the migratory breeding owls, owls, that they are no longer migrating back to the north to breed. They're sticking around. And they're breeding with the residents. So both anecdotes and my data show that these are places that we suspect that's what's happening there. So if you're wondering how the heck you catch a burrowing owl, this is your answer. These are essentially like small mammal traps. Owls are kooky. These burrowing owls are kooky. They are up at night and during the day. When do they sleep? They live in these burrows in the ground. They are active through the diurnal cycle. We mostly catch them at night because in many of the places it's really hot where they live. but also because the way that you catch them is to put these traps that have two little doors on each side, cover up the traps. You place them at the opening to their burrows, and you get the owls either coming or going from their burrows. So that's most likely a whole bunch of nestlings that were just coming out, some fledglings at this point that were coming out. They're all captured together. It's really fun to work on fledglings. They're all idiots. There was something I was going to do. Let me just do it. Okay, so burrowing owls are also, they have this really interesting defense response the nestlings do to prevent predators from getting them. That it's kind of daunting when you're a researcher working in the middle of the night in the pitch dark. Many of the owls that I worked on were in artificial burrows, and the way the artificial burrows work is they put like PVC pipes under the ground, and there's usually an opening that's just a bucket from Home Depot, and you pop the bucket, and you have to, there's maybe 3 feet of space around in their burrow, and you have to reach all the way down to get to the chicks like this. But sometimes you hear this sound. Now what I'm going to play is first going to be a rattlesnake, and then it's going to be a burrowing owl. They mimic rattlesnakes. I was supposed to play it there. For some reason, we couldn't figure that out. So occasionally you're reaching in there and it sounds like a rattlesnake's in there. Funny story about that is I was told that no burrowing owl researcher has ever been bitten by a rattlesnake. It's not something to worry about. And then I gave pretty much this talk at a burrowing owl conference last year, and I learned that a burrowing owl researcher was just bitten that year. So I'm glad I missed that experience. All right. So overall, what I did was to sequence a full genome. So all the genetic data, all the DNA that are in the cells of one of the burrowing owls, I sequenced a 50x, which just provides a really high resolution reference to be able to compare the rest of the data to, so we can know where in the genome the rest of the data are. And then with the rest of the samples, I sequenced 214 burrowing owls to what's called low coverage, and this, now we know this is very experimental. Back when we did this, was ultra low coverage, which is essentially less than 1x, which means that we only have a representation for each one of those 214 owls of a single representation of each base pair in the genome, each nucleotide in the genome. And what this does is it allowed us to be able to sequence a whole bunch of burrowing owls, get genetic data for a whole bunch of burrowing owls all at once for not terribly expensive. And we could get the overall picture of what's going on with the burrowing owls. And then we learned that we need to do more, which is just how science works. The data sets that I'm going to be referring to, the analyses that I'm going to be presenting, are based on, depending on the analysis, somewhere between 2 million and 10 million single nucleotide polymorphisms. So it's a ton of data. It's a ton of data. Here's what we found.
Drill down to individual owls.
Now we can. With this data set, it's really best to ask questions at a population level. You really need the combined data all together. But I'm going to follow up talking about what we're doing now. And now what we're doing is getting more data so we can know more about each individual owl. Okay, so this is what we found. This is a representation of genetic structure at the top there. I don't want to harp on that. The PCA here is more informative. Now remember that triangles are migratory populations, circles are resident populations. So each one of those circles is an individual owl. Common colors are from common sites. And what this is telling us is that we have this structure. So this little line, this is just how genomic data comes out in PCAs. This little line of samples that are all together, suggesting that there is closer genetic relationships amongst those samples, that there's essentially it's its own population there versus this one over here versus this one over here. And all of the migratory sites are right here. They're piled up on top of one another. That means there's no genetic structure. That means that burrowing owls, whether they're in British Columbia or North Dakota, they're just as likely to be able to share gene flow. Owls can move completely across their migratory range. But in their resident breeding range, there's a great deal of genetic structure. There's less movement between these populations. What's more, I compared a measure of genetic structure. It's called FST, really common statistic for being able to diagnose genetic structure among populations. And what this is telling you, so on the left here are all the migratory sites together. On the right, all the resident sites together. And what this is showing you is that genetic structure is much greater among those resident breeding sites. Over here is a comparison of geographic distance versus FST versus that genetic structure. And there's two different signals here. Among the migratory sites, that's what this line represents, is a flat line. There is no relationship between genetic distance and geographic distance. Again, meaning that owls are just as likely to move among sites in British Columbia as they are to move between British Columbia and North Dakota, or New Mexico for that matter. But among the resident breeding sites, there's what's called isolation by distance. So there's a really strong relationship between geographic distance and genetic distance among the sites. And what this implies is that the owls possibly can move between these resident breeding sites, but they're more likely to move between sites that are closer in space. And that's significant. That's significant for a couple of reasons that we're going to be talking about later on, including the management of the species. So I'll put a pin in that and come back to why that matters in a minute. Among the resident breeding sites, I also found that there is much higher levels of inbreeding. Again, this is suggestive of reduced gene flow. So FIS is a statistical representation of inbreeding. So you can see higher inbreeding levels in these resident sites versus the migrant sites. Again, that matches right up with this story of really high gene flow, really a great deal of movement between the migratory sites, versus those resident sites, is suggesting those resident sites, there's not much movement, there's not much gene flow, and perhaps there's some inbreeding to think about in going forward. This breaks down site by site. Each of these colors over here are resident breeding sites. All the resident breeding sites had much higher inbreeding than the migratory breeding sites. Okay. So the next thing I wanted to do was to ask questions about selection between these migratory breeding sites and these resident breeding sites, to try to understand if there's any sort of natural selection that might be either driving this migratory behavior or otherwise underlying this migratory behavior. So what I did for this was to compare a pool of all those residents together versus all of the migratory sites together, and to, again, look at genetic structure, but here looking at genetic structure across the genome. So on the x-axis from left to right, each one of these numbers represents a chromosome in the burrowing owl's genome. And for this, I wanted to pull out sites in the genome that were exceptionally differentiated, which could be suggestive of selection. So pulled out those sites, the top 0.1% loci and the top 0.01% 50 kilobase windows, and did an analysis called a gradient forest analysis. That's a type of regression analysis to compare this the genetic information at those sites versus a whole bunch of environmental variables to try to see if there's any sort of associations between the genetic data and the environmental data. And I found really strong associations with several environmental data points, including one that captures barren land. Burrowing owls live in open areas. They're known as a grassland species in many places, but in other places it's open deserty areas. mean temperature of the coldest quarter, mean temperature of the coldest month. Hey, the resident, the migratory birds live in a place that gets really cold. So these are things that are differentiating the birds environmentally, and potentially also driving some differences in their alleles as well. So once I figured out that there was a significant relationship between these environmental variables and these particular sites in the genome on particular chromosomes, I then tried to determine if there are any particular genes that might be in windows around these parts of the genome to see if there's anything that makes sense, essentially is the way it works, that might help explain the differences between resident and migratory breeding owls. So what happens is this gene ontology analysis. You get a whole list of genes that are in these regions. And then you do an analysis to try to figure out if any particular gene pathways are overrepresented amongst these genes. And sure enough, what I found was the lipophagy pathway was overrepresented. Lipophagy is processing of fats. We know that birds build up a whole bunch of fat before they migrate. And so perhaps this is evidence that these migratory birds are significantly differentiated at these particular genes for building up fats. Kind of matches the story a bit. So, to my questions, I got answers. There are different gene flow patterns based on their breeding behavior, these different phenotypes. The migrants are panmictic, which means that there's gene flow completely across the migratory breeding range. The residents are in isolation by distance, so there's limited gene flow between essentially like neighboring sites. There's higher inbreeding amongst the residents, and there seems to be some evidence for relationships between environmental variables and the genetic data. All right. So this beggars additional questions, of course. I didn't point it out, but there was the evidence up there about this potential migratory switching, the possibility that owls were migrating south and then finding the conditions really nice where the residents are and sticking around and possibly breeding with the residents. But my data wasn't able to really tease that apart reliably. The only other data that's out there was very much anecdotal, some observational data. It's kind of a funny paper that was put out that suggested this was happening. But people on the ground, people who work with the owls, were suggesting this was happening. But now we know from this research that we can differentiate those resident breeding owls and the migratory breeding owls. So we knew that we would have a tool to be able to tease this story out. Is migratory switching real? Are birds sticking around and breeding with the resident owls? Furthermore, in the future, and there's been a couple things I'm going to point out that have already been done to collect more genomic data, to get higher resolution data for the individual level owls to be able to ask questions that I couldn't ask. So one of those things that was already done, my collaborators up at San Jose, there's a project called Head Starting, which is sort of like a captive breeding project, but what they do is they go out and gather up newly fledged owls, bring them into containment, because that is the time of life. As I said, fledglings are pretty idiotic. They're really easy to catch. So that means they're easy to catch for coyotes as well. So this Head Starting project up in San Jose, they bring these owls in and then keep them during this time of their life, re-release them later on. And they were also starting to getting into pairing of the owls as well. So my collaborators working with that project went out and differentiated using genetic techniques over the winter resident breeding owls versus the migrants who were here for the winter. And they were able to determine that, yes, indeed, there was some evidence that some of those wintering owls those wintering migrants, did stick around and bred with the residents, which is really kind of interesting for a lot of reasons. That population in San Jose is really small. The figure I showed before shows that there's a great deal of inbreeding there. So, hey, maybe that's a source of owls for them in the future, that they can grab one of these really healthy migrants and keep it around and try to breed with their population so they can boost the genetic diversity of their population. And guess what? They did that. So the same collaborators went on to use the genetic data, higher resolution genomic data for individuals, to try to figure out how to pair individuals to eliminate evidence of inbreeding. So when animals undergo inbreeding in a population genetic way, we refer to it as non-random breeding, but really we're talking about breeding with close relatives. And the problem with inbreeding is what, students? For you guys, what's the problem with inbreeding? Recessive alleles, deleterious recessive alleles. So what happens is when organisms get highly inbred, their genomes become less heterozygous, they become less variable, they become more homozygous. And when inbreeding happens, these regions of the genome called runs of homozygosity build up that are places where genes that are essentially really bad for the fitness of the animal, deleterious alleles, can be expressed because they are homozygous recessive in their genotype. So what this figure represents from this study, on the y-axis is, these are band numbers, but each one of these is an individual. On the x-axis is a representation of runs of homozygosity in the genome for one of the chromosomes. I don't know how long this is, but it's a couple 100,000 base pairs. In these individuals, where this dark spot is, that's a whole run of the genome where both chromosomes have the exact same variants on them. And that's bad. That's bad for the organism. It's a way to represent the possibility that these deleterious alleles might be expressed and affect the fitness of the owls. So what they did was to pair up one of these individuals that was expressing this run of homozygosity. I shouldn't say expressing in this, just had this run of homozygosity with an individual that did not. And then they went back after they bred. They showed that by pairing up an individual with a healthy genome with an individual that did not have a healthy genome, they could produce offspring that had healthy genomes. So they can pinpoint, increase the genetic diversity of the population in a way that we can never do before. Before it was just, let's go grab some owls from some other population and hope that it helps. Now we can say exactly which individual we should be putting together to increase genetic diversity in the population. Not only that, but it's been shown that telomeres are the ends of our chromosomes. One of the things that happens as we age is our telomeres get shorter. It's one of the causes of aging. It turns out that that's a good representation of fitness. I'm an older person too, so no offense. So they show that the individuals that were paired, their offspring actually had increased telomere lengths. So this is a relationship between this telomere length and the fitness of these animals. They increase their fitness, at least theoretically. So it's a really cool study that they did. But I'm continuing to work on burrowing owls with those collaborators to try to increase the genetic information that we have, population by population across California, to inform future translocations of the owls, to try to do some more of this really assertive kind of conservation to bring their populations back, to prevent the continued extirpation of their populations along the fringes. So I'm hoping to get information that will allow us to say just how far we can move the owls from their homes. Because the possibility is that owls that are out in the Mojave might have adaptations that differentiate them from owls that are up in San Jose, and we might actually produce offspring that are less fit if we pair those, if we translocate owls from the desert over to San Jose. So the projects that I'm working on now are trying to answer questions about selection in greater detail to be able to understand how likely moving additional owls in the future will be. So that'll allow us to do further mitigation in the face of continued urban expansion, moving owls away from their homes so that their homes don't get destroyed, that they don't get destroyed as their homes are destroyed, boosting small populations like up in San Jose, trying to understand the populations that need help, need to have translocations occur to increase their genetic diversity and reduce inbreeding, and possibly reestablish populations. That last one is just me. So hang on to that idea for a minute. So we have already worked with collaborators to increase the number of sample sites in California from the 4 that I showed before up to 15. This data set includes a whole bunch of samples. I'm working with a longtime researcher on burrowing owls named Courtney Conway. Those are his pictures that I showed before of the owls in cages, in traps, owls in cages, owls in traps. Courtney has been working on burrowing owls throughout their Western North America range for 30 years. And so he's got samples from 20 years ago. Do I have to stop? Okay. So we have a whole bunch more samples. We're increasing the coverage of the genomes. So instead of having essentially one representation of each nucleotide in the genome, we can have 8, which allows us to be able to say more about what's going on in their genomes. We can get higher resolution analyses, looking for more evidence for adaptive variation to explain differences between our populations, and get higher resolution analyses of inbreeding. Now, to that last point about reestablishing populations. One of the ideas that I have is, I pointed out earlier, this is from the same slide from earlier, that of all the 6,500 breeding pairs that are left in California of resident breeding owls, 4,000 of those pairs are in the Imperial Valley. This is what it looks like where those 4,000 individuals are. That's the Salton Sea up there. This is all desert on the outside of these polygons. Notice all this green in the middle here. Somebody know what that is? It's agriculture. It's irrigated agriculture. So this is an area, and I pulled this out of environmental consultant report. These environmental consultants went out and tried to find all the burrowing owls that are breeding out there. And each one of these dots is a breeding burrowing owl. So there's 4,000 breeding burrowing owls out there, and it is flush. And I don't have any evidence for this. I just have a suspicion that there's no more space for them to get to there. So my suspicion is that they're producing a whole bunch of offspring that are leaving this area and not really finding a place to go because there's not as much rich habitat outside of that particular region. So boy, wouldn't it be nice to be able to go out there and harvest a whole bunch of burrowing owls and move them around. That's my idea. I want to promote that idea more. Put a pin in it. We'll get to it in just a second. Just a second. Yeah, I got a really good idea about that. Yeah.
You may be heading in this direction, but the nestlings that are, have the improved genetic structure, are they relocated out into the wild? Is that happening?
Yeah. Yeah, absolutely. That head starting program, it's not a captive breeding program. They are pairing the owls and putting them back into habitats up in San Jose that are currently being protected.
Out in the hills somewhere, I suppose.
I don't know directly. I know one of the sites is right next to Google, because look it up. There's a New York Times article about how Google had this thing going on where people were supporting all of these outdoor cats at the Google offices, and the cats were killing all the owls. And so these researchers actually managed to get Google to pay to put up a fence around their habitat to keep the cats out. So that's a place where they're recovering. Yeah.
How much pressure can you put on the pesticides and herbicides they're using in agriculture? That's going to affect the genomes.
I believe other researchers are working on that. It would certainly be interesting to know. Certainly, I mean, a direct impact. These owls, I didn't mention at the beginning, they're mesopredators. It's actually kind of interesting. I've seen what their prey look like. Working on one owl out in Nevada. The males hunt while the females are brooding in the burrows, and the males will bring back prey, drop it outside of the entrance to the burrow, and then call to the female. And that's kind of how we keep track of them at night. So I've come across lizard, multiple scorpions, and I believe a mouse just like laid out as a smorgasbord for the female to come out to eat. So certainly they eat insects, they eat arthropods. It's a big part of their diet. Arthropods are directly impacted by those pesticides. So it's a direct impact there. But the indirect pollution, the chemicals that they're exposed to, I'm not sure. So it's clear that there's a lot of food there for them. So the way I think about it is you know, a bunch of people scoffed when they were thinking about the irrigated agriculture, and yes, we're overusing our water resources in places like this. But for the owls, this completely changes the habitat for them. So they're doing really well out there. Yeah, it's a good question. If they get past their fledgling years, I think owls can live up to 10 years, but they are very much in the middle of the food web. Coyotes love them. It's actually a big problem that I've been thinking about, because I've been thinking about where to relocate owls, and where you site their artificial traps is really important, so that the owls can stay vigilant and see the coyotes coming, essentially. So they are very much in the middle of one of those classic food webs underneath coyotes in particular.
How far from, I guess, before our expansion of agriculture, what do you think their range and populations were before we got hold of the agriculture?
I mean, certainly there weren't this many owls out there. There couldn't have been as much energy out there for the owls to be able to consume as there is now. I mean, they ranged all the way to the coast. We had owls. There's the Monterey, what is it called? The Atlas, the bird atlas back there. When was that done, you reckon? Like
92, I think. Well,
oh, thank you for that.
88 to 92.
And so there were burrowing owls reported at the Salinas River refuge, I believe, in that book.
No, those were all no breeding on the coast.
Okay.
Breeding is in the lower Salinas Valley in the southeastern part of Monterey County, particularly Lone Oak Road and Wild Rose Road, still have breeding populations.
Oh, they do?
Yeah, but other known populations like the Salinas Airport had a populations. Those disappeared after the Atlas, so in the last 20, 30 years.
And the really frustrating thing, I've researched it and worked on these owls for 10 years. It doesn't seem like we know exactly why there's been this recession from the coastline, but up and down the California coast, there's been complete recession away from the islands, away from the coast. Some of the samples that I have from Courtney Conway are from populations that don't exist, and that's just from 20 years ago. So places like North Island and San Diego, yeah.
Is the San Joaquin Valley a site for them?
For sure. There's yeah, absolutely. Central Valley in general, yeah.
Yeah, I'm just wondering why what's so great about the Imperial Valley? There's so many other agricultural places in California. Why aren't there 4,000 burrowing owls and some of the other?
I don't know what it is about the Imperial. They apparently are doing really well in these. I haven't been out there, and I need to get out there to see it. They're apparently doing really well in these ditches, in the rows along the fields. I'm not sure what it is that allows them to do so well there and not in King City, not in Salinas. I'm not sure. Yeah. That's right. Yeah, so my suspicion is that this is a completely changed habitat. Birds are going there and overwintering, and there is some connection between this cue to migrate and their environment. So that cue to migrate, the environment is communicating with their genes and saying, "Hey, this is a good place to stick around. Why make that migratory journey to?" Who knows if there's still any grasslands up in Manitoba that you can get to? Yeah.
All the breeding populations in Monterey County are in grasslands that are foothills and very open and grazing land and large ranches. The Salinas Valley is too dense in our constant agriculture.
Oh, okay.
There's no even patches of bare ground of any size. So it takes just a huge open grassland. It's one of the reasons we call the King City grasslands an important bird area. It's formally designated that as by the Audubon Society Cal Audubon about 30 years ago, because of that habitat that's good for them and the prairie falcons and a few other things that require wide open spaces. Then the Salton Sea is I've been there a lot.
Okay.
A mixture of open desert patches, open mesquite patches, open sand dunes, and patchy irrigation. So there's plenty of open space.
Okay, that makes sense. All right. So, of course, I want to thank my funders for facilitating our research, including Pasadena Audubon Society that gave me money every year that I was down at UCLA. I want to thank my family for support. I was hoping that they were going to be here, but I was explaining to Shannon back there that this little girl, this time of day, should be running circles around this place right now. Watching. Oh, thank you. My son is apparently online watching while my daughter's asleep. Okay. So just real quick, I want to finish up by talking about some research that my students did this past fall. As Amanda mentioned, this past fall, I taught the first iteration of bird biology at CSUMB Monterey Bay. With this class, I really wanted this class to have a research component because that's my background. That's something that I can offer students. I wanted to encourage students to get outside. It's something I do in several of my classes. I want them outside, want them working with here birds, and thinking about science and learning about the process of science. So before I taught the class, I was going around asking a whole bunch of people questions about what are the needs? What do we need to know more about here in this area? I've lived in this area a bunch, but I haven't really been involved with Audubon just yet. I haven't learned about all the things that are going on here. And then my fellow faculty member, Jarek Bergsma, said, "You should talk to Amanda Priest." And so I reached out to Amanda and I said, "Hey, I want to get students involved with research in this project. Do you have any ideas?" And Amanda had some ideas. I went and had an hour-long meeting with Amanda, and just a lot of amazing ideas here. And then what I did was to just allow the students to self-select. I just offered them a bunch of these ideas as possibilities and allowed them to choose them. And then I gave them zero guidance whatsoever. Is that close to home? And allowed them to struggle and try to figure out the problems as they went along. But they did some pretty cool things. A couple of students, including Alexander here, was inspired. They were inspired by this incredible Point Pinos count that happens in November, right? Yeah. I wouldn't be surprised if Alexander's in this picture somewhere. So these students wanted to work with this data to try to understand any sort of environmental components that might explain these trends over time that have been observed in the Point Pinos count. And I'm going to kind of fly through these and just talk a little bit about what everybody did, more so than any sort of final conclusions, because all of these projects need more work to have some final conclusions. But I think it was a worthwhile experience for my students. So what this figure is showing are the counts of a variety of species that are observed from Point Pinos during this count over the course of 9 years is what the study was. And they really honed in on the Pacific loon, which is this one here, because it's really bouncy. You know, there's been some big differences in Pacific loons over the years, and they really want to try to figure out if there's any sort of environmental variables that also were equally as variable over time. So they looked at sea surface temperature, and that was kind of suggestive. There was some sort of matching that happened between sea surface temperature. They looked at wind speed, they looked at wind direction. So that's the work that they did. Some other students, including Max, who's here today, were inspired to try to understand the impacts of this new campground that's being built over on Fort Ord Dunes in the state park. Amanda suggested that, hey, you know, it's really cool that we're getting this new campground, but anywhere there's human infrastructure, crows show up, and crows eat everything. So Max and his collaborators went out and created these transects from the campground inland to try to measure the current rate of observations of crows. And on the x-axis here is essentially at the campground. They couldn't get all the way to the campground. They had to go up to the barrier and use the binoculars to see as close to the campground as they could. So essentially at the campground, 1 kilometer away, 2 kilometers away, 3 kilometers away. And they found that, in fact, the crows weren't really that common just yet. So it'd be fascinating. I believe Amanda has researchers who are currently doing more following that up. Some other students were inspired by this idea, again, by Amanda, to compare bird diversity on beaches, also to collect data on human behavior, essentially. If people were bringing their dogs to beaches that weren't supposed to have dogs. I would love to know what to do about that when I observe that myself, because I certainly see it a lot. How often people allow their dogs off leash on beaches that they're not allowed to be off leash. But essentially, they found this relationship between the numbers of dogs on beaches and a lack of birds. So Carmel Beach had the fewest number of birds compared to like Marina Beach. Audubon directly supported a different project that a couple of students were inspired by to go deploy these devices called audio moths that could collect sound data they deployed them for 2 weeks down in Big Sur. And the idea was to try to figure out if there's any evidence for the presence of spotted owls, an endangered species that it's a hot button topic online. If you guys haven't seen, it's a big thing happening in Oregon right now where they're going to go out and slay 500,000 barred owls to protect spotted owls. And they collected 1,000 hours of audio recordings and not nary a bit of recording of spotted owls, not just yet. Have you heard any spotted owls yet?
I just retrieved the audio models like last week. I haven't run it yet, so TBD.
So maybe in the future. All right, here's where I said put a pin on it. So the idea of where to reestablish burrowing owls. We have grasslands right here, very small ones on Fort Ord. So an idea that I am working on is the idea if we learn using the genetic data, the genomic data, that there's no great adaptive differences between, say, owls in San Jose or owls in other nearby populations and the Imperial Valley owls. Could we reestablish a burrowing owl population up here in Fort Ord? So some of my students, a couple of whom are here today, just working with environmental variables, created a habitat suitability map to try to say something about how much suitable habitat there is for owls. Again, all of these works can use more work to get to something that would be like a publishable product. but at least just using climatic variables. They clearly didn't do anything with an urban footprint or anything like that, or an agricultural footprint. They showed that, so where this white area is on the map, it's really good, suitable habitat, at least using the climatic variables for burrowing owls. So, hey, maybe there's a possible a lot of places out there to bring burrowing owls into back into this area. Just a crazy idea. Yeah.
But any of the other protected, like the
I'm betting that there would be some bloodletting in meetings to make decisions about this between people who are concerned about tiger salamanders. Would a burrowing owl salamander? Yeah. So it's certainly something to think about. And my students actually did bring that up as things to think about for this possibility. And I'm just like, hey, it's an idea. I would love to have some burrowing owls in my backyard. I live right off of Fort Ord. So that's it. So those are the projects that they did, and I thought it was cool work, and Audubon supported a lot of it. So I just wanted to make sure that I presented to you guys, and I'll finish with some pictures. Doing scientific research with kids around is difficult. They're very curious about what Dad's doing on his computer. But I'll finish there and take any questions that you guys might have.
What was your experience? And do you feel like now that you have the capability and the higher coverage that you're finding your work to be easier or more reliable?
I certainly hope so. What we had back then when I did this analysis was a really good understanding of chicken and zebrafinch. So I got to compare those genes to chicken and zebrafinch, and with the wealth of new genomic data that we've gained in the 6 years since I've done this, I am looking forward to seeing what's available. But I don't have an answer for that just yet. And actually, that's going to be the responsibility of a grad student at Colorado State, not mine. Can't wait to see what he finds. Yeah.
I think reestablishing the burrowing owl population is an awesome idea. A lot that would go into that. What would be? Have there been any projects? One that where they tried to reestablish burrowing owls in areas? How did those go? And then two, how do you account for like inbreeding? Maybe happens if they are just together.
Well, I mean, if we have genomic data, then we can avoid you're thinking about after we reestablish them, if they continue being small. That is something to think about for sure. I would think that we see with the isolation by distance that it's not like there's no gene flow going on between their populations. They're more likely to share gene flow between neighboring populations. So possibly there might be gene flow between places like here and San Jose and other nearby sites. What was the other part of your question?
Well, have there been any?
Interesting. In the state of Arizona, and there was if you guys get National Geographic, just a couple months ago, there was an article about this group called Wild at Heart. And you may have seen me thank Wild at Heart up there for getting samples from them. Arizona is kind of the Wild West for species conservation. They've allowed this particular group, Wild at Heart, to move burrowing owls across the state willy-nilly without any sort of understanding of the consequences. That was also one of the sites where we saw really high inbreeding. So they're pairing owls poorly because they're not using genetic information to make those decisions. Here in California, the San Diego Zoo is doing really amazing work trying to understand what leads to translocations that are successful versus the ones that aren't. What they found is that if you translocate to a new area, you also should translocate a whole bunch of California squirrels as well. Yeah. So the best success was those with those California squirrels. It sure seems like there's a lot of California squirrels around here, though. Oh yeah, there you go. Yeah. Yeah.
Tree squirrels.
Yes.
Is there any models for spotted owls that might involve a little less slaughter?
Oh, can I invite you to come to a debate in my class in the fall? Is there another model? Not one that I'm aware of. Not one that I'm aware of. I can't imagine any other system for removing the animals that are removing the spotted owls. It's a tough call. It's heartless. I'm not going to be the one to do it, but here's what I do know. I worked on the black cap vireo in Texas when I was with the U.S. Geological Survey. My collaborators down there worked on Leakspell's vireos. The thing that helps their populations return, the thing that we've seen boost their populations is removing brown-headed cowbirds. And there are really, I mean, on Fort Hood in Texas alone, when I was working there, my understanding is they had removed hundreds of thousands of brown-headed cowbirds. And you know what I mean by remove. So this is a very effective conservation technique. And brown-headed cowbirds are not hurting for populations, hurting population numbers, yeah.
I'm from the Northwest and the lumber industry is part of my career. And its habitat is what it's all about. You can try to, look at, focus on parts like the spotted owl, which was a real focus there for a while. But because the forests are being decimated, and it's still being decimated every day. I go up to the Northwest all the time, and the log trucks are hauling out the fields. And you're not going to try to fix parts of the populations if you don't leave the forests.
Yeah, reestablishing good habitat is the conclusion that my students definitely talked about in their debates.
It takes care of itself.
Real quick, I think an announcement before we end. Kelly mentioned, actually Don mentioned King City Grasslands Important Bird Area. Monterey Audubon has just recently published a nice little flyer about that burrowing owl that's represented on the back. I just looked on eBird while I was back there, and 5 days ago one was seen out in Snow Valley, a little bit further east than the King City Grasslands Important Bird Area. Feel free to pick one of these up at the back. And then for those of you that are also the same boat as Kelly, where you're like, oh, what to do about off-leash dogs and all these things, so these lovely brochures.
Oh, I'll take that.
Your dog or friends, or you can just say, "Oh, hey, a park benches over there. There might tickets. There's something coming up. Figure out where to go and have fun at the beach legally." So just wanted to plug in for these right at the back of the room.
Thank you, guys.
For some snacks too, if you want to hang out for a little bit.